Much for being with us today. As you can see, he's got a couple of other jobs. He's the Deputy Dean for Teaching, and he's involved with agri-sciences at Stellenbosch, but he really loves these microorganisms. And so today he's going to tell us a little bit more about what we need to know about these bugs and what they have hiding up their sleeves. Prof, over to you.
Thank you, Linda, for that kind introduction. It's always nice to have you as the chair introducing someone. You just feel so good after that specific introduction. So let's continue with my presentation so that we can start with that one. So today I'm going to talk to you about how to identify the problem, but actually about the resistance of microorganisms to sanitizers, specifically in the food industry. And it's interesting to note that my slide is not moving. There's moving. So it is interesting to note that there's emerging trends that's happening nowadays regarding microbial contamination in the food establishment. It's up by 150%. And we all know about the listeriosis outbreak and what that did to the food industry. But we can see that we've learned a lot of lessons from that outbreak, and that most of the contamination from microorganisms, including pathogens like Listeria monocytogenes, is actually associated with your production environment and not always with the raw materials that you're using in that specific product. So there's a new trend that's actually happening that's shifting from testing the final product to more investigate into your environment and what you're doing in that specific food processing environment. It leads to a point that you will have to ensure that your environment is going to be clean and your environment is going to be safe. Then I can move to the next screen. Seems that I'm not going to use it from my side. So the food industry is facing a huge problem regarding microorganisms due to the fact that they are actually starting to overcome all the factors that can kill them. So they're no longer susceptible to the sanitizers and the disinfectants that we use in the food industry. They're becoming clever, and you will now know by now that I will always say that the microbe will have the last word. And it seems like these organisms are actually trying to adapt into an environment so that they can survive. And therefore, we see a global rise in the resistance of these organisms. So if you look at sanitizers, we need to look at the definition of those word like a sanitizer. So it's an antimicrobial that kills or inactivates at least 99.9% of all bacteria, fungi, and viruses. So that means we call it a log3 reduction. So if there's 1,000 organisms on a surface, after you've adding the sanitizer at the correct dosage and the correct time, there should be only one left on that specific surface. So when you talk about these bacteria, fungi, and viruses, we call them microbials, microbes, microorganisms, bugs, whatever we think about it. But at the end of the day, we need to start to control them. So the resistance towards a sanitizer of these organisms is a growing concern, and you can see it in some of the industries. We saw that quite clearly with the listeriosis outbreak, that there was a group of sanitizers used in the industry, and Listeria were becoming hugely resistant to that specific sanitizer. But I'll touch on that a little bit later. So these organisms then survive the final step of your cleaning process, and therefore gives them ability to persist in food factories. Although the number of organisms surviving is low, they become then the predominant organisms in your factory and can cause a lot of problems if you don't use the correct concentration and the correct contact time of the sanitizer. So what is the difference between these type of products? So the cleaner is basically where you aid soil removal. So it basically removes the dirt off the surface, but also removes some of the microorganisms that's not attached to a surface, but it does not actually kill them. Then you look at the sanitizer, and that is reduce the number of bacteria to a specific level considered safe by public health organizations. And then you have a disinfectant that kills basically the bacteria, the fungi, the viruses with a log 5 to log 6 reduction, but it does not have any effect on your endospore forming or endospores in that specific environment. Then you have a viricide, and that's specifically to kill viruses like the COVID-19 that we do have. And then we have a sterilant that actually eliminates everything to zero, zilch, nothing that we can talk about. So it's important to note that there's a difference between a sanitizer and a disinfectant and a cleaner. And if somebody say they've got the one in all, you have to think twice about it, because all of them do a specific function. So what is the effect on microorganisms? So on this Petri plate here, on your left-hand side, we can see basically white discs that's on there, and on each of these discs is actually a sanitizer with the specific microorganisms, and you can see clear zones around that disc that is there. That means that the organism that is that ivory-colored bacteria on the Petri plate will not be able to grow in the presence of that sanitizer. So if you look at the Petri plate on your right-hand side, you can see some of these discs actually still have clear zones, while the others, the bacteria, can actually go up to the disc that is there. So that is how the bacteria becomes resistant to the sanitizers. With the green arrow, you can see that specific sanitizer is still an effective sanitizer after multiple uses of the sanitizer to that specific bacteria, while the other one that's been indicated by the red arrow is actually the one that's becoming ineffective sanitizer, and therefore your sanitizer will not have any effect on that microorganisms, even if you use it at the correct concentration and at the correct time period that you have to do there. So the driving force behind the sudden emergence of sanitizer resistance can be largely attributed to the abuse and the misuse of sanitizers in the industry, either by not using the correct concentration or not allowing the correct time for the sanitizer to be effective. So how does the spread of organisms start? So as with antibiotic resistance, antimicrobial resistance is following more or less the same pattern. So it is a selective pressure that is on the organisms, and you must remember these organisms are in a survival of their own. So they will basically create all different activities, mechanisms to actually overcome this. So you can either have antibiotic or antimicrobial resistance that's passed horizontally between the organisms by means of genetic material that they do have, or we can have a vertical one, where the one that's already been resistant just multiply, and they actually give rise to more organisms that's going to be resistant. Now, some of these genetic elements is actually situated on a plasmid, and the plasmid is actually a piece of extra DNA that can quickly be acquired by other bacteria of the same species, and even interspecies they can sometimes change these plasmids. So let's look at the resistant mechanisms. So there are 4 resistant mechanisms that's been used by the bacteria to actually overcome the effect of the sanitizer that is there. One of the most common one is the one that I've indicated there on your left-hand side, the top corner, is basically the efflux pump, and that is where the sanitizer will get into the bacterial cell. It was supposed to start to basically disrupt some of the genetic material, but then the organism will recognize the sanitizer or disinfectant that's in the cell, and effectively pumping out that sanitizer from the inside of the cell to the outside of the cell. The second one is actually where we have enzymes that are actually designed to basically modify the antimicrobial, the sanitizer, and that antimicrobial enzymes originate usually from a plasmid, and that is the extra piece of DNA that is loose from the bacterial chromosome, and they start to basically make sure that these sanitizers will be ineffective to kill the organism. Then from the same plasmid, there can be some other enzymes that's been developed that we call the antimicrobial degrading enzymes, and that enzymes then actually breaks down the antimicrobial to a point that is not going to work. So these 2 mechanisms is usually associated on a plasmid, and that plasmid can then be interchanged between some of the organisms that does not have that specific extra piece of genetic material. The 4th one is actually from the bacterial chromosome, and that bacterial chromosome is actually targeting to modify the site of the sanitizer to work. So it's usually on the outside of the bacterial cell. And by modifying the site of attachment of the sanitizers or the disinfectants of antibiotics that is actually there, they cannot attach to that specific site, and therefore they cannot actually eliminate or kill that specific bacteria. So the use of sanitizers becomes very, very important because we totally depend on sanitizers for hygienic food factories, and the resistance of the sanitizers to food-related and disease bacteria is at the moment not looking good, because a lot of these foodborne organisms are actually building up resistance to a wide range of sanitizers that's been generally used in the food industry, and is escalating to become a global crisis. But not only for normal organisms in the factory, specifically to the foodborne pathogens, as we've seen with the listeriosis outbreak that we had. So microbes such as bacteria, we know, are living organisms, and they're evolving all the time due to the pressures that's there where they need to grow and develop. And it's important that they are going to adapt to these specific environments. So the presence of the sanitizer and the microbes are killed, or they survive, or they basically carry on with the resistant genes. So the survivors will then start to replicate and quickly become then the dominant type. That's been shown in the figure at the bottom, where the light-colored pinkish organism is the one with the resistance, and the other bacteria are basically the normal organisms. And if you use a sanitizer, you will kill off all the blue cells, and then the resistant one will survive. And then that will be the one that's going to multiply and to grow and become the predominant organisms in your factory, ending up with a group of bacteria that's actually going to be resistant, and that you will find it very difficult to get out of your factory. So we've seen this specifically from Listeria monocytogenes. There's some numerous articles that's been published regarding the use of different sanitizers to eliminate or reduce the risk of Listeria monocytogenes. And what was clearly seen in an article published a few years ago is there was a steady increase in the sequence type number 6, the same one that's been associated with the listeriosis outbreak in South Africa, over the years in the Netherlands. And as they discovered that there's an increase in the sequence type number 6, they were looking into much more detail about the organism and the genetic makeup of the organism. So what they did is to look at specific genes that will link them to become resistant to specific sanitizers. And they discovered that this ST6 had the specific efflux protein that pumps out the quaternary ammonium compounds from the inside of the cell to the outside, called the EMRC gene. And together with that EMRC gene, there was also another gene that was actually there for the organisms to survive and increase their capability to form a biofilm. So you had basically two genes specifically on the genetic makeup of the ST6. First one, that they are actually pumping out the sanitizers so that they can survive, and the second one, that they were able to form some biofilms on surfaces. So even if you use any type of sanitizer, in this case benzalkonium chloride, that's been extensively used in the industry, it then led to the buildup of these highly pathogenic organisms within the food factory. So if you look at QACs, there's about 7 different generations of QACs, and I'm not going to go into the detail of all the QACs that is actually present there, but the bacteria is not only resistant against benzalkonium chloride, it's becoming resistant to all the 7 generations of QACs that we do have. So with the multitude of sanitizers available to the industry, ranging from quaternary ammonium compounds, which are the most common that's been used, we also have peracetic acid that's been used, lactic acid that's been used, and sodium hypochlorite that's been used. And it's clear to see, and if you look at specific pathogenic organisms, I'm just referring to Listeria monocytogenes, because this is the one that's been studied quite widely to see how organisms adapt into food environments. You will see from the international literature that it's got a long-term effect on the outcome of these pathogenic organisms. But even if you look at Salmonella, E. coli 0157, or the sugar toxin producing E. coli, or Campylobacter, that you will find that there's a resistance towards some sanitizers that's been actively used against them. And together with that, if you look into the genetic makeup, for example, of Listeria monocytogenes, you will see that there's an increase in the specific genes that are associated with resistance to sanitizers, specifically the QACH1, that's specifically for quaternary ammonium compounds, and then you have the EMR genes that's actually there to form some efflux pumps to pump the sanitizer out of the bacterial cell. So we're sitting with the situation that a normal bug is going to become a superbug, and a lot of people are talking about antibiotic resistance because of the abuse of antibiotics in the public health systems. But even in the food systems, we're going to create some superbugs because of the abuse of sanitizers and the misuse of sanitizers in the industry. And one of the things that is actually quite important is to always make sure that personal hygiene will be catered for, and you should spend a lot of time educating the people of the benefits of personal hygiene. So there was a study done in Ireland a few years ago, over a 5-year period, specifically looking at the impact of sanitizers on Listeria monocytogenes in a meat processing environment. And it's quite interesting to see that although not all of the organisms are making use of the same techniques for the resistance, around about 50% of all the organisms that was isolated, specifically Listeria monocytogenes, showed one or other form of resistance towards a sanitizer, not only benzalkonium chloride, or basically the QACs, but they also had some transposons, and the transposons is actually a group of genes that is actually responsible for resistance of sanitizers. They also had, some of them had the plasmid, and the plasmid, as I've shown earlier to you, are actually the ones that can transfer the resistance of the sanitizer from one bacteria to another. And of course, yes, they also have the efflux pumps. So not all of the bacteria had all three of them. Some of them did have all three of these, the transposon, the plasmid, and the efflux pump. But one of these groups of genes are then responsible to ensure that there will be resistance. So biofilms is important because everybody studies the organisms in its free living state, a state where it's easy to kill the organisms. But as soon as the organism starts to form a biofilm, it becomes very difficult to actually treat them. Linda, if you can go to the next slide, where we actually show that how a biofilm do form. So firstly, we see that some of the bacteria will have an idea, and the idea is actually to attach to a surface, and then they will start to have the initial adhesion towards that specific surface that is actually there. The second step is actually where they start to basically multiply in that environment and then actually produce some extracellular proteins to attach them as like a glue that is actually there. The third one is actually where they will find that they are now matured and they're starting to recruit some other organisms, and most of the time it's not only the same organism like Listeria, but there will be a multitude of other organisms from E. coli right through to other organisms that will then start to accumulate in that specific biofilm. And then lastly, as with any type of any idea, there will be some of them that are not happy with the lockdown situation that they have now at this moment in this specific biofilm. So they want to go out of lockdown 4 into lockdown 3 and 2, and then they will move then and form a new biofilm at another stage. So it becomes important that it's a protective mechanism for the organisms in a food factory. So what we've done at Stellenbosch University is that we've actually studied some of these biofilms to see what is the effect of sanitizers on these biofilms. And on your left-hand side, the one with the light blue and purple is actually where we start to inoculate the organism. It was a Listeria monocytogenes that we've isolated actually from a food processing environment to see that, yes, that blue one that is actually there is actually where the organism is forming a biofilm, and you can see it specifically from that curve that is actually there. The same is for the orange one that is actually at the bottom. You can see it's starting to form a biofilm, and then that line that is going down is due to the ESCOM power failure, so it's not that the organism has actually died off. So let's go to the next slide, where we can actually show you what is happening if there's a sanitizer added to the Listeria monocytogenes. So we used here a quaternary ammonium compound sanitizer, and the left one where they indicated the D, you can see that there's a growth of the biofilm, and suddenly as we add the QAC, the growth then basically stops, the organism is dying off. But within a matter of 50 hours, you will actually see that there's another growth that's happening. So these group of organisms that's been then surviving the sanitizer is starting to grow out again. But as you then use the same sanitizer, you kill them off as well. If you look at the one on your right-hand side, the P one is actually the same, but just a different organism. And you can see as you add the sanitizer, the organism is dying off, but within a matter of hours, that organisms are starting to grow again. And then you have a much shorter interval where you have to add the sanitizer. So the one in the P is much more resistant towards that sanitizer as the one that's actually left on your left-hand side. And even if you see that from the tubes that we're using after we've been studying them, in the outflow tubes, you can clearly see, and I hope you can see that there are patches of organisms that are attaching to the surface of the plastic that's been there. And that is after you've actually at numerous times have added the quaternary ammonium compound. So these organisms that are actually in that tube of the outflow must be one of the most resistant organisms that you will find in your industry, and that is why it's important that you make sure that you get rid of your biofilms together with a good sanitation method that's in place. If you look at the next slide, it is actually important that we're using also some other sanitizers. The left-hand one is the QAC from another company, and the other one is a non-QAC or quaternary ammonium compound free sanitizer. And with all of them, you see that initially you can kill them off, but within a matter of hours, there's some new organisms that are actually forming in that same environment. Some of them are much quicker than the others, so the longer it takes for the organism to recover from the sanitizer, the better it's going to be. And you look, for example, at the one at the right-hand side, the purple and the blue one, you can see after the second round of sanitizers, we had basically a total drop in the counts, and therefore almost 100% kill of that specific sanitizer. So it's important to know your sanitizers and what your sanitizers can do and how you can actually to control them. So even Salmonella is becoming resistant to hypochlorous acid. Many years ago, we did a study on the resistance of hypochlorous acid to Salmonella in a poultry abattoir, and it's quite interesting to note that if they become resistant to the HOCl that's actually there, they also start to produce the heat shock proteins. So if you have to use heat later on to kill them off, they become more heat resistant due to the fact that you've been using hypochlorous acid right in the beginning. So how do we control the microorganisms? Firstly, we need to keep them out of the food factories. Second one, we need to have agents that can actually kill them. And if we will not be able to kill all of them, we need to make sure that we will prevent the multiplication of these bacteria, viruses, whatever we're going to want to eliminate, and control the growth of them. So those are the three basic things that you always need to keep in mind. So why is it that we have ineffective sanitizers? First one, because we have rushed cleaning practices. We don't stick with the concentration. We don't stick with the time. Disinfection of any food facility is a dynamic process. It's not a once-off. So from you start with production till the end of production, before you start with the cleaning process, you need to monitor basically what is happening on. And you have to have specific trained staff to ensure that you will have adequate disinfection and sanitation that's taking place. And one of the things that become more and more important is that hygienic design of equipment and factories is starting to play a huge role. So if you don't design your equipment in such a way that you can easily clean them, you will always find that they will be resistant of organisms for the sanitizer that you've been using. So, in conclusion, the food industry faces a potential collapse if infectious agents are no longer susceptible towards the disinfectants or sanitizers used, and therefore we'll see an increase in foodborne pathogens, and foodborne outbreaks will become more and more common and will be a threat to food security if we're not paying a lot of attention to the sanitizers and disinfectants that we do use. So with that, I want to thank you, and as always, like Louis Pasteur said, it is the microbe that will have the last word. Thank you very much for listening. If there's any questions, please do ask me the questions that you want. We've also spent a lot of time doing some research on UV and UV, the impact on different pathogens. Between myself and Professor Ziggy, we've published some articles on UV. So if you want to have a more academic impact and see what we've done, you're more than welcome to ask for reprints of that. But we're also part of the Center for Food Safety, and the Center for Food Safety is a nice initiative that we innovate through collaboration. We do high quality science. We basically respond almost immediately to emerging risks and opportunities, and we train students so that they can have an impact and they can serve the industry. So